Okay. I like it. I wish my kids quieted down as quickly. I'm always like, hello. Okay. So I did some accounting, and it's really awesome that we're going to get through all of this stuff by the end of Friday. Because I was a little worried since we lost a lecture. But we're good. Okay, so today we're going to finish up the last bit about motor proteins and talk about regulation of the cytoskeleton and how basically in response to some kind of stimulus you'll have signaling which will affect how the cell moves or what it does, and so just the effect of the cytoskeleton on cell behavior. So first, some business. The extra credit was what happens when dynein reaches the minus end or kinesin reaches the plus end of microtubules. Does it just detach? Does it get stuck there? So some people sent answers that were like, it's not clear, I couldn't find anything. And I would agree with you. It wasn't very clear. Some people sent an interesting physics paper that said something about how it detaches. It's cold in here, huh? How it detaches and takes a subunit with, which was interesting. And then there were some viable options, but nothing that convinced me. And it sounds like it's not completely clear, or that I just need to talk to some new works on motor proteins. So basically, if you sent a viable answer, meaning an answer that answered this question, because sometimes people like to send me other answers to other questions, but if you answered this question and gave me a reference that was legitimate, I'll give you the point, because you tried. Sometimes there's no answer, or sometimes it's not clear. So it's not like you need the right answer. Okay, right. So my thoughts, though, in my research was that since motor protein activity is often regulated by phosphorylation, it makes sense that it might detach, because if you need to phosphorylate in order, like for one of the myosins, for it to assemble into a functional thick filament, that seemed like there was that, and there was another motor protein that's also regulated by phosphorylation. And it made me think that, well, if there's an active and an inactive conformation, then why not just make it inactive when it's done? But I don't know. I never found anything supporting that. Okay. Last thing, call for exam questions. Anyone who wants to try to submit a question for the exam, due by 5 p.m. tomorrow. I meant to do this on Monday, but I forgot. So tomorrow, 5 p.m. 5 p.m. That's the other thing. People send me extra credits well after the deadline. I don't look at it. It has to be, so if you send it at 5:05, I won't look at it. The other thing is sometimes people like to share questions. Like I'll get the same question from multiple people. I won't look at those either. So you can work on questions together, but have your own that you're submitting. Like I don't want 5 of you guys to submit the same question, expect the same points. What else? I think that's it. Questions about any of those fun things? Good. Okay, so we learned about the components of the cytoskeleton. We learned about the proteins that regulate the cytoskeleton, and in recitation, you've thought about that very deeply, hopefully. We learned about proteins that organize the cytoskeleton. We started to talk about motor proteins and how motor proteins are regulated. So what are 2 examples of things that could regulate the activity of a motor protein? I can think of 2, I guess, small molecules that might regulate the activity of motor proteins. One I just talked about. Huh? Well, no, small molecules that regulate the activity of motor proteins. Yes? Right, so phosphorylation was one. That was the one I just, myosin, right? ATP. Well, okay. You know, that's an obvious one, right? Because you need ATP. Wasn't what I was looking for, but it is a right answer. Yeah, ATP. If you don't have ATP to be hydrolyzed, you won't have movement of the motor proteins. And what's another thing that regulates the activity of motor proteins? Think muscle contraction. Calcium, right? Calcium. I heard someone say calcium up front, right? So without calcium, you'll have a motor protein and it'll be there, but it won't be able to associate with the actin. So remember when I talked to you about how we should start thinking about grouping things? That was an example of how you might think about something. Like, we talked about many different ways of regulating motor proteins, right? So it's worthwhile to think about grouping things like that. And so we started actually to talk a little bit about the cytoskeleton and cell behavior with muscle contraction, right? Because that is regulation of the cytoskeleton and it has an effect on the behavior of the cell, right? Maybe. Does anyone agree? Yeah, I see some people nodding. Good. Just keep nodding. Then I'll feel like I'm getting through, getting the message out. Okay, so today and Friday we'll talk about structures made up of cytoskeletal components, structures that are permanent, and structures that are made as needed, right? So it's an example of a permanent structure made up of cytoskeletal components. We didn't learn it yet, but you did do your reading this weekend. And you have taken a lot of cell biology by now, right? I don't mean just this class, but like starting at freshman year. Permanent components made up of permanent structures made up of cytoskeletal components. Someone give me one. Yeah. Centrioles. Yep. Well, they are permanent, so there's one. But think about structures rather than that. So a centriole is a structure in the sense that you have multiple microtubules emanating out. Yes. But think. Yes, that's one. Let's try for another one. Yes. Flagella, right? Do flagella go away? Or do they come in response to a signal? Do sperm, like they're normally don't have flagella, but they grow them quickly when they're needed? No. Sorry, it's so funny to me. I'm just imagining. Right, so cilia and flagella, I have it right up there. So, and then things that are built as needed, mitotic spindle, right? That's a big one. Is the mitotic spindle a permanent structure? No. Okay, good. And cell protrusions, right? What do I mean by cell protrusion? What does that mean to you guys?
Like microvilli?
Like microvilli. Yeah, but microvilli tend to be permanent structures, right? Pseudopods. So any sort of protrusion that is involved in anything from endocytosis through cell movement, right? So that's where some edge of the cell moves out one way in the direction that it's moving, or the edge of a cell will come in with endocytosis. Okay, so I just wanted to get you guys thinking about what we're going to be talking about. Okay, so this is where we ended the other day. Cilia and flagella. These are motility structures that are built from microtubules and dynein, right? So flagella in sperm and protozoa, important for swimming, right? Cilia, this is the wave-like movement of, in your gut, right, intestinal cells. And where else are cilia? Your lungs, right? Wave-like movement of adjacent cilia can move organisms through liquid and also move fluid over cells in a tissue, like mucus in a respiratory tract, eggs in an oviduct. So the core or axoneme of cilia and flagella, it's composed of microtubule doublets. Remember, there are these rings. See, this is a cross section arranged in a ring around a central microtubule doublet. So it's a whole bunch of microtubule doublets. And the way they work is through ciliary dynin, and it binds to a neighboring microtubule. So here's the head. This is the motor domain, right? And here's the tail. And this will be bound to another microtubule up here. And the ATP hydrolysis results in walking to the minus end of one microtubule while remaining bound to another, which results in the bending motion. So this is how it looks right out of the book, right? So basically, dynin move along one microtubule while remaining fixed to another, and depending on and that results in the bending, and it's dependent on these proteins that link the two microtubules. So they're bound together, and when one so it's stuck here and then it's walking here, and that results in it going like that kind of, but it goes like this because they're they're attached to each other. So this takes us to the quiz question that we had this weekend. Dynin functions in the bending of flagella and sperm and protozoa. The bending of the flagella occurs through the following mechanism. So there's a lot of words here, right? And really, you could have gotten rid of most of these, right? So dynin moves along one microtubule while remaining fixed to the neighboring microtubule. That was the answer. And you could have gotten rid of most of these, right? Dynin moves along actin. No. Dynin has two motor domains. One moves along one actin filament. No, right? Because if you got actin there again. Dynin has two motor domains. One moves along the microtubule towards the plus end, while the other moves in the opposite direction. So if they're both completely sliding against each other, then you're not going to have that bending. So you need to have it fixed on one end. Questions about this? Okay, so if you had a defect in ciliary dynin, what would be the outcome? So think about something that has this dynin, and what would be the outcome if it didn't work? I'm going to stand here until someone gives me something. My watch isn't working. Did you guys see that they have these my husband just showed me these watches now that are basically smartphones? Have you seen this? They're like that big. You can get your email, and it's a watch. It's like, takes you back to like the old superhero comic books. Dick Tracy, yep. Anyways. Do you really want to email? I was thinking that's really the worst of both worlds. Like, already we had to like become blind by reading everything on smartphones. And no one wants a big, bulky watch. So now you've got a big, bulky watch, and you're even more blind because everything's smaller. So I'm wondering how that'll do. Anyways, defects in ciliary dynin. Oh, okay. So ciliary. So this is that. Are they moving? Well, they're moving. Yeah, the stereocilia. Yeah, I didn't think that wasn't the one I was thinking about. So deafness maybe. So the cilia in the ear could affect hearing or balance. Yeah. Okay, in the lungs, more prone to infection. Okay.
I would say like emphysema, when you smoke, doesn't it destroy your cilia and then you get smokers?
Okay, so again, emphysema, lungs, right? So when you smoke, it destroys your cilia. So, well, but we're talking about the motor protein.
Yeah.
But if that would probably destroy everything there.
Yeah.
Yep.
Cause digestive problems that cause the cilia to not be able to move in the intestinal lining.
Okay, so digestive problems if the cilia couldn't move in intestinal lining. Okay. So what about dynin's in flagella? Infertility, that's a big one. I do wonder if that's an obvious, like it's obvious logically, but do people check for some mutation when people come in with non-modal sperm? Swim in circles, yes. Okay, good, good. Okay, so they are rooted in the cell surface. So cilia and flagella are rooted in the cell surface by basal bodies, which are similar to centrioles. So what do you think is common between the centriole and the basal body? What do you think is a common component other than microtubules? Do you remember what a centriole is? Yes. Yes, so that's what I was looking for. Gamma Turk, right? So that's what nucleates and grows out your microtubules. So chances are you're going to have that in your basal body, right? So do you think that the microtubules in cilia and flagella are dynamic, highly dynamic? Or do you think they're stable? I hear people going, stable. And what kind of protein do you think might be affecting their stability or preventing the possible dynamic nature of microtubules? Yes? So some kind of plus N capping protein, maybe? So those of you who already did recitation this week, that should have been fairly obvious because you guys just had to go through all your proteins and what they do, and you should have automatically thought about that. And I'm guessing you all did, but just were feeling shy today. Right? Who's feeling shy today? Who doesn't want to put, see, some people feel shy. I think it's important. It's important to know how you're feeling. So today's not a good day for shy people to do karaoke, right? Who does karaoke stone cold sober? Yeah, see? Oh, good! Someone does. Okay. Forgot to close the door. People are so good at not coming late that I forgot to close the door. I tend to remember to close the door and people are always coming late because they bother me. Okay, so there are structures that are constructed when they're needed, right? So myofibrils, cilia and flagella, those are stable structures, right? They're always there. Others need to be constructed when they're needed in response to an internal or external signal. So what are some examples? We kind of talked about earlier. Things that need to be, cytoskeletal structures that need to be constructed in response to an internal, from within the cell, or external, from outside the cell signal. Yep.
Pseudopods.
Right. Pseudopods. Structures associated with creating a leading edge on a cell. What else? Come on, we said this already. Everyone should be jumping. Okay, a few people saying mitotic spindles. Right, so those are the most obvious structures that need to be constructed when they're needed. So this should be taking you into the thinking of regulation. What regulates when these structures are made, right? So microtubules and motor proteins are required for the construction of the mitotic spindle, right? So that's what we just said. Right? So the interphase microtubules need to be disassembled. So here's interphase. Oops, let's turn off the light. Leave it like that maybe. Right, so here's interphase, see, one centrosome, microtubules emanating outwards, right? And here is what's happening here? What kind of strange process is this? Right, so this is the mitotic spindle. You have duplication of your centrosomes. What phase is that? Metaphase, thank you. So this actually happens in less than an hour. So this is I would say this is, yeah, we'll leave it that way. I think there's some more microscopy. So I would say that there's some kind of regulation involved that's going to cause this change, right? It's not random, is it? No. Interphase microtubules undergo rare catastrophe. something to think about. And this actually bothered me with the people's answer with the motor proteins about how they detach and shorten taking subunits with them. I was like, well, I wonder, like, does that mean that they're more dynamic than, I don't know. Anyways, so they undergo rare catastrophe. What kinds of regulatory proteins do you think are likely to be bound to interphase microtubules? Rare catastrophe. Yes.
Proteins that are capped on the ends.
Right, so some kind of capping proteins, proteins that bind to the ends that are stabilizing. Yep. What kind of proteins will you probably not see? So let's say, yeah. Katanin, probably not. I'd say that's a fair assessment. Yeah. Remember, katanin chaps up your microtubules like crazy. Not good. What else do you think you would not see? You don't need to give me an exact name. You can give me a type of protein and its function that works on a test. If you didn't remember katanin, but you said protein that binds to microtubules and chops them up, you'd get it right. Catastrophe factors, right? So these are these proteins that bind to the ends, sides, ends, and disassemble. So probably not many catastrophe factors bound to microtubules and interphase. Okay, so how would you answer this question in the lab? So you have some hypotheses, right? I just heard hypotheses. I heard katanin, I heard catastrophe factors, I heard also, so that shouldn't be there, and I heard capping proteins should be there. What would you do? What would you do in the lab to evaluate if you were correct? What would be your experiment? I know, I'm asking such difficult questions today. Yes? Huh? Okay, so you think we've formed a hypothesis here, right? So the question is, what kinds of proteins bind to microtubules and interphase, right? And so we've come up with a list of proteins that we think would be bound and things that we don't think would be there. But now we need to test it. It's a hypothesis. It's not fact. We haven't seen this. Now, the obvious thing is to look in the literature because someone's clearly done this before. But it's like 1978. I don't know. This is probably a lot more recent. It's recent. It's much more recent. It's 1995, and it hasn't been done. And you're doing your PhD, and your advisor says to you, I wonder what kind of proteins are involved with microtubules during interphase. They seem to rarely undergo catastrophe. And so you come up with this list. So what would you do? Like, you just submit the list to Science for publication? No. Yes? Oh, so there's one way. Make knockout cell lines that don't express these proteins and see the effects on microtubule dynamics. That's one. Yep.
With like antibody fluorescence for those and see where they're actually bound. So for those specific proteins.
Right. So do immunofluorescence using antibodies against these proteins we just listed. Right. If we're right, they should be there in interphase. And maybe what would we compare it to though? So they should be there during interphase and not when. Yes? Mitosis. I mean, there's really only so many options. If I'm saying interphase or the other, right? There's only so many phases to the cell cycle. Right. So compare to mitosis. Do live cell imaging and watch to see what proteins are where when. Or do knockouts and look to see the effect on dynamics. I think both of those would have to be done because you need to show things a million times. Okay. Good. Who is like inspired to do research from these questions? Who wants to drop out of being a bio major? Okay, well, it's too late. You guys are about to graduate. Okay. Motor proteins in the mitotic spindle. Okay, so we keep talking about mitosis and how important microtubules are in mitosis and the formation of the mitotic spindle. So there's also motor proteins involved in formation of the mitotic spindle, more in formation of the mitotic spindle than in actual chromosome segregation. And so this is a nice figure that shows you your mitotic spindle, only three sister chromatids. What organism is this? Does anyone know an organism that has 3 chromosomes, or 6, depending on diploid or haploid? I have no idea. So I'm guessing it's just for simplicity. You know when people draw mitosis and they just draw 1 chromosome? Anyways. Okay, so lots of motor proteins. Motor protein, all these motor proteins. You've got motor proteins, and it's all about positioning the mitotic spindle, right? Because don't forget, if you just have 2, you've got your centrosome duplication, right? And they just hang out there, and they're not oriented together in any way, then they're not going to be lined up and in the proper position to then separate those chromosomes, right? So a big part of it is making sure they're in the proper place. And there's a whole bunch of motor proteins involved. And you do not need to memorize them all. I shouldn't have had to say that, but you know, some people really love to memorize things. Okay, so there's 3 classes of microtubules that make up the mitotic spindle. There are these astral microtubules that they're basically helping to center the mitotic spindle, and they're pushing against the plasma membrane. And then there are the microtubules that actually bind. These are the kinetochore microtubules that actually bind to the kinetochores on the chromosomes and the sister chromatids and help to separate them. And then there are what else are there? There are these interpolar microtubules and so this is all motor protein here, right in the middle. And they help to kind of center everything also. Okay, so I finally figured out a way to include this figure. So that's enough about mitosis for now. It's coming back in the cell cycle, and it's going to come back in gory detail for those who can't wait to take 409 with me. Who's taking 409 with me? Whoa! It's going to be like a reunion. We'll all bring our husbands and wives and show pictures of our children. And we'll brag about what they're doing or not, or we won't say anything about them. They're not doing. Mine's in the best preschool. I've never been to a reunion, but I hear via movies. So that's okay. Big picture. So I wish I had shown this like on day 1 of the cytoskeleton, but I think it's appropriate here too because we're going to start talking about regulation of the cytoskeleton, and one of my favorite examples is cell movement. So this is awesome, right? So the big picture. I know it's really easy to start thinking about these individual proteins and how will I remember them. Dr. Ehrlich told me to draw this stuff, and even though I had to do it in recitation, it still didn't quite do it for me. So here's another one. So here's your cell, here's your plasma membrane, and it's awesome because this shows everything, right? So if you start, where's A? I don't know where A is, but let's start here. Oh, here's A. So you've got this pool of actin, right, bound to profilin. Actually, what they should have done is had also like a thymosin bound pool, but that's okay. And look at that. It exists in an arrow shape. Isn't that amazing? So you've got this pool of actin, and it's hanging there, and it's just ready to be added to whatever it needs to be added to. So there's some kind of extracellular stimuli. There's bacteria in the area. And the cell's like, we must go this way to the ceiling. We must go north. And so basically it signals activation of ARP, right? Remember we learned all about ARP. And that signals activation of this, or creation of this kind of web of actin, right? So that's C. Where's D? Where's D? Yeah, there you go. So ARP binds, you have elongation, and then there's E, which is somewhere over here. And so you've got the growing filament that's pushing your membrane forward, right? The whole point here is you're chasing your bacteria. And then you've got capping proteins, whatever. And then at the back end, we get to the back? Okay. ATP hydrolysis, remember? So you've got your T-bound new filaments, your D-bound old filaments, right? And so remember, we've got cofilin in the back end that's binding to the ADP-bound old filaments and disassembling them. And those subunits go into the pool of profilin bound. And actually, I didn't tell you this, but in this picture it shows you profilin catalyzes the exchange of ADP for ATP. So it makes it into, it's ready to be recycled and added to the front again. And then it follows the arrow back to the top. There's no real arrow, in case you didn't catch that. But I thought this was so awesome. And actually some people even pulled it up in recitation on Monday. And I had just found it the other day. And I was like, this is the best. The big picture. So when you find yourself trying to memorize all these little bits of detail, take yourself back to a picture like this, and you can see how it all works together. Any questions about what I just talked about? Okay. I know you guys probably at night while you're sleeping say to yourself, big picture. Big picture. Okay. So here we're going to start talking about cell crawling. Actin-myosin and these regulatory proteins that we just looked at, they all work together to promote cell crawling. Right? So you'll have, here's your actin cortex, so your cortex of your cell, and that's where your actin web, your ARP-mediated actin web starts to grow, right? And so it pushes forward. This is the lamellipodium, and it's crawling along the surface. And you've got two things that are happening, right? So you've got this protrusion, and so that's this actin polymerization at the plus end, which protrudes the lamellipodium. And then you also have, so all this unpolymerized actin is moving in this direction. It's diffusing across the cell in the direction that the cell wants to move. And then you have contraction actually happening at the back end. So there's two things happening. You've got actin movement. You've got this web-based actin forming at the front via ARP, nucleation by ARP. And in the back, you've got this contractile actin and myosin too. So what kind of nucleating protein do you think is in the back end? I'm telling you, there's contractile actin working, bringing up the rear, right? Yep. Formins, right. Formins are mostly involved in formation of contractile bundle stress fibers. Exactly. And they're working with myosin back here to kind of help bring the rear forward. So actin polymerization at the plus end. So this would be the plus end. Drives membrane protrusion. And filipodia, lamellipodia, pseudopodia, these are all different types of membrane protrusions. Here's okay, so more microscopy. Here's ARP. ARP is green, and actin is red, and green and red make yellow, kind of, in microscopy. And what you're seeing here is here's your cell, and the leading edge is full of ARP and actin. Notice no ARP in that rear part. Probably, if you stain for formins, you think that they would be back here? Probably. And probably not up here, right? Because you've got that contractile actin in the back and the web kind of actin in the front. Right, so ARP binds to the sides of actin filaments, which forms this 2D web of actin filaments. And it's a treadmilling that drives the web that drives the protrusion. So treadmilling, well, I'm telling you about ARP, right? So remember we learned about treadmilling, which is really just the addition at one end and the subtraction at the other end that makes the whole filament kind of move forward. So something that must be removing something from the back end, right? What do you think that is? We got ARP helping things grow at the front end of the leading edge, and at the back end, you're disassembling your actin. So there's more actin that can add to the front. We'll look at some pictures. Right, so we have nucleation at the leading edge and depolymerization of old filaments behind the leading edge by cofilin. And this maintains and drives unidirectional motion. So here's your leading edge, and here's the back, right? And the green is cofilin, and the red is actin. And so you can see, this is a magnification. You can see right here you have actin only. This is the leading edge, right? And right over here you've got all this cofilin associated with actin. And that's cofilin binding to the old actin and dismantling it. Does that make sense? Does everyone believe me? People think I lie to you. I would never lie to you. It's true. So how is the activity of cofilin driving this treadmilling process? Well, what is it doing to help drive the movement forward? Yep. It's right. So it's essentially creating subunits and monomers that can be added to the front. Where are those subunits and monomers coming from? From the polymer. Is it where though? Like where in the leading edge? The end, right? So the back part, remember cofilin binds to ADP bound actin. So it's taking apart those old actin filaments and creating a pool of actin subunits that can be used to build new actin, pushing the cell forward. Anyone want to add to that? Okay. Every now and then I realize I haven't looked at the clock in a while. Sometimes it sneaks up on me and I'm like, oh, we're done. But we're not done. Okay, so this is what I was just talking about, right? So so this is the direction that you're moving, right? Is that right? Something's not right. Oh no. Wait a minute. Sorry. Yep. This is the direction that you're moving. So here's your new actin, and you've got a capping protein, and you've got your ARP, and then in the back, you've got cofilin binding and disassembling, and then and then those actin monomers follow the arrow and move to the front and bind to ARP, and then assemble new actin. So just reiterating what we've shown about 5 times. Okay, here's a question for you. ARP and cofilin are involved in directional movement of cells. They likely function in the following way. ARP nucleates at the lead. Okay, raise your hand if you vote for it. ARP nucleates at the leading edge while cofilin breaks down old actin to free up monomer for actin assembly, driving a treadmilling effect. Okay, good. ARP nucleates actin at the leading edge while cofilin brings in free monomer to the foramen whiskers, driving a treadmilling effect. Okay, good. ARP disassembles actin at the leading edge while cofilin nucleates old actin at the centrosome. Okay, I'm really bad at writing wrong answers, aren't I? ARP nucleates microtubules. Okay, so you guys should all be getting A's on my exams because I'm so bad at writing bad answers. Right, so the answer was A. ARP is nucleating actin at the leading edge, and cofilin is breaking down the old ADP-bound actin to free up monomer for actin assembly, and that's what drives the treadmilling effect that pushes the cell forward. Okay, so I just told you something about contractile actin in the rear, right? So myosin 2 is associating with actin web. I don't know if web is what I want right there, to pull the trailing edge forward. So in this image here, you've got actin stained in blue, so it's around all the edges, and myosin in pink. So actually myosin is more associating back here. So that contractile actin associated with myosin helps scooch the back forward. That's the scientific word, scooch. Okay, so how's this all regulated, right? So I'm telling you what happens, but something needs to turn things on and off, right? So now we're going back into the world of signaling for a little while. The Rho family of proteins is involved in actin organization to generate cell polarity in response to extracellular signals. I keep telling you these things are happening in response to some kind of signal, right? Nothing's random in the cell. Well, I guess something's random. I don't know. But most things are not random in the cell. So there's some kind of signal that's signaling the cell to migrate. And the Rho family of proteins is involved in this regulation, right? So the front and back of a migrating cell have different structures, right? We just talked about this, right? In the front, you've got this ARP induced web of actin, web of actin. Oh my Lord. Okay, is the semester over? No. Okay. And in the back, you have this formin-induced contractile actin. And so something's got to be regulating the fact that those are assembled in different places, right? It's not just like, it just so happens to be that way, right? So if you look at myosin 1, so you've got your leading edge over here, and there myosin 1 is only at that end in green, and red is myosin 2, and it's only in the rear, right? So Rho proteins are part of the RAS superfamily, right? So what does this tell us about the possible functions of Rho? Yeah, it has a GTPase. It's a GTP binding protein. It functions to do what in signal? What do these GTPases function as in signaling generally? Switches. Right, they're molecular switches. What do switches do? Okay, can I show you what a switch does over here? Oh, turn things on and off, right? They turn things on and off. That's not very scientific, but it's true, right? Yep, so they trigger cascades. They turn things on and off. Right, the RAS superfamily of proteins, of which Rho is a member, are GTP binding proteins that function as molecular switches, right? So when they're GTP bound, they're active, and when they're GDP bound, then they're not active, right? They're inactive. On and off. This is like, what are the other molecular switches we've spoken about? Have we talked about any other molecular switches in this class? RAN. What's RAN involved with? Nuclear transport. What else? What other molecular switches? Come on, we like to group things together. I'm going to put it on the final exam. Anything else? Huh? RAB. RAB. What's that involved with? Vesicular transport. What else? RAN, RAB. Yep. Right, so RAN was all about nuclear import and export, exactly. The directionality. Yeah, the directionality. What else? No, that's regulation of the activity. So I heard GAF. GAPs and GAFs regulate these molecular switches. So the other one was RAS, right? And so now we have RO. RAN, RAB, RAS, RO. They're all the same, but they function in different places to do different things, right? Okay. RAC, RO, and CDC42 are members of the RAS family. Based on this information, these proteins are likely to have the following function. GTP binding? Who said no? Involved in signaling? ATP binding? No, consistently no. So I would say it was probably A and B, maybe? A and B, or D. Yes. 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 Or D. A and B or D. Really, D. Okay. Okay. RAC and RO and CDC42 are members of this RO protein family, which is a member of the RAS protein family, and they're involved in signaling pathways that affect actin organization. Right? So we talked about this receptor, and the receptor sees some kind of signal of like bacteria, and then there's signaling that results in activation of ARP and WASP and actin web formation and movement of the cell to chase the bacteria. So this picture is showing actin staining with different members of this RO protein family because they do different things. Okay, so here's a normal cell. You stain the actin, and it's just got a little bit of actin around the edges, and it's not there's nothing going on. When you have activation of RO, so this is a cell where they constitutively activated RO, and you get stress fiber formation. That's these lines. And they're staining for actin only here. And if you constitutively activate RAC, you get lamellipodia formation. And if you constitutively activate CDC42, you get Philopodia formation. So that suggests that these families, these molecular switches within the family of the RO protein family, right, they all activate what? What's different about them? They do different things, so they probably activate different signaling pathways, right? Right? Okay. God. We've got like 2 minutes left. Can you guys power through? Can you do it? Are you up for the challenge? Okay. I see someone saying no, so I'm going to give them a hard time. Okay. How do RO proteins affect actin organization in response to an extracellular signal? Right? So you can draw this out without me teaching it to you, right? You've already learned signaling, right? So all you need to do is show that something's being activated, something's being phosphorylated, something's being activated, something's being inactivated, blah, 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 activation of ARP, blah, 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 activation of formins. Remember? It's so generic, you can just draw it out. The blah, blah, blahs, those are like triangles and squares. Okay. So WASP proteins are frequently activated by CDC42 and RAC. So here's a nice picture of how this all works. You've got receptors. This is from the company I used to work for. But they do pretty decent pathways. So if you're ever trying to figure out a pathway, it's a good way to go. You've got receptors, and then you've got these RO family GTPases, and they could activate all these different things. You can get, here's your stress fibers, your contractile actin, your web, your ARP-mediated actin web formation. So activated WASP binds to ARP to enhance ARP nucleating activity. Does this sound familiar to anyone? Who's saying no? Raise your hand if you're saying no. Who's in recitation last week? Every single person in this room should find this familiar. We did a whole paper about this, right? Does everyone remember this? So E. coli tear recruits ARP and WASP. You guys were like, why is she teaching us this? E. coli tear recruits ARP and WASP through neck to induce pedestal formation, right? This is what's going on here, right? So in this, remember this figure? This is the pedestal. Your E. coli is the green, and your tear receptor is your red, right? And then over here, you're looking at EPEC plus actin. Here you're looking at EPEC plus WASP. Here you're looking at EPEC plus ARP. The whole thing forms a complex, which is important for pedestal formation, important for forming a web of actin that protrudes the plasma membrane up, right? Okay, I'll leave you alone. Go away.